So my name is Anna. I am a bioinformatics software engineer in the Myon Lab at the Icahn School of Medicine at Mount Sinai in New York. And today I will be presenting two related tools for transcription regulatory analysis. The first being ChEA-KG, which is a network-based transcription factor enrichment analysis tool. And then I'll show you how we extended ChEA-KG to create ChEA-KG-TS, which is a time series transcription factor regulatory analysis workflow. So as most of you probably know, transcription factors are proteins which bind directly to DNA to control gene expression. There are about 1,639 known human transcription factors that together form a transcription factor regulatory network. And since transcription factors can regulate the expression of other transcription factors, activation and inhibition arrows can be established to denote their regulatory relationships. So the figure on the right here is a classic example of a regulatory network from the sea urchin and mesoderm, and it demonstrates how these regulatory networks are highly complex. So there are a variety of both computational and experimental methods that have been developed to infer and reconstruct these regulatory networks. But before I get into that, I want to first quickly describe a little bit more how these networks are structured. So the nodes in these networks are transcription factors, and then the edges are the regulatory interactions between the transcription factors. So I have an example here showing the transcription factors SPI1 and GATA1, but the edges have a direction which assigns the source and the target roles. And then they can also have a sign which indicates the effect of the regulation, namely activation or inhibition, as well as a weight which can be used to describe the statistical significance of the likelihood of the interaction, as well as also the strength of the interaction. So one way of constructing these networks is by performing transcription factor enrichment analysis. So very broadly, this method involves predicting the likely transcription factor regulators for an input gene set from the results of differential expression analysis. And so the background transcription factor target gene set libraries that are used for the enrichment analysis can be assembled from a variety of sources, including data from published ChIP-seq studies, positional weight matrices, or gene-gene co-expression correlation analysis. So the figure here is summarizing the various sources that are used by a tool developed by our lab called ChEA-3. So the libraries are shown here. There are 6 total. So there are several other transcription factor enrichment analysis tools that are summarized in this table. The first tool is the subject of this presentation, and it was actually just published last week in the web server issue of Nucleic Acids Research. So this tool, ChEA-KG, along with our other tool, ChEA-3, are both highlighted since they were tools developed by our lab. But the important takeaway here is that only a few of these tools provide interactive network visualizations with signed interactions using networks that include all approximately 1,600 human transcription factors. And those are the categories that are highlighted in yellow in this table. So now I'll explain in more details what ChEA-KG is and how it expands on our previously published tool, ChEA-3, to fill this gap. But first, a little more detail about ChEA-3. So ChEA-3 is a popular transcription factor enrichment analysis tool from our lab. Its development was led by former MD PhD student Alexandra Keenan. And as I mentioned, the enrichment analysis is based on 6 background gene set libraries of transcription factor target relationships. And these are collected mostly from ChIP-seq and co-expression experiments. So the figure here is showing the individual transcription factor coverage of all of the background libraries. But in total, ChEA-3 covers 1,632 known human transcription factors. And the website URL, as well as the link to the publication, are here if you'd like to read more. But ChEA-KG combines data from ChEA-3 with another tool developed by the Myon Lab called RemeGeo. So the development of RemeGeo was led by a former lab member that some of you may know, Giacomo Marino. He's now pursuing his PhD at Yale. But RemeGeo is a search engine that serves about 180,000 human gene expression signatures. and these are uniformly processed from studies that are published in the Gene Expression Omnibus. So RomaGeo contains a wide variety of up and down gene sets, and the link to the site as well as the publication are here again in case you want to read more. Sorry. So to create ChEA-KG, we took a subset of those RomaGeo gene sets, specifically gene sets that had clear control and perturbation conditions, and then we systematically submitted those to ChEA-3 for transcription factor enrichment analysis, while aggregating the edges between the top 10 inferred transcription factors and the genes that they regulate in each gene set. And then because we know that the genes were up or down regulated, we can use this to infer the sign of the interaction as well. So after constructing the initial network, we pruned it to only include edges that were observed more frequently than would be expected by random chance. And so then this filtered network is uploaded to a knowledge graph database where it's served via the web server application ChEA-KG. So below is the link to the site as well as our recent publication. But the final network has 1,559 human transcription factors and 131,000 signed and directed edges. So the central feature of ChEA-KG is this network-based enrichment analysis tool. So after performing transcription factor enrichment analysis with ChEA-3 based on an input gene set, it produces a subnetwork that shows the enriched transcription factors connected with the edges from the background network. So these edges can be used to, and subnetworks can be used to identify master regulators and suggest regulatory mechanisms. So for example, in this network, HMGA1 in the middle here has a high concentration of edges and is therefore likely the master regulator of this specific transcription factor module. So one of the neat features of ChEA-KG are the ChEA-KG atlases. So these atlases are context-specific collections of transcription factor regulatory networks for 4 different themes. So first we have normal cell types, pan-cancer tumors, drug mechanisms of action, and changes in human tissue due to aging. So for the cell type atlas, this is displaying regulatory subnetworks for 131 cell types from 14 different tissues. The Cancer Atlas has subnetworks for 69 subtypes, which we identified from the 10 tumor types of the CPTAC 3 cohort that you all are familiar with. The MOA Atlas, or Mechanism of Action Atlas, has subnetworks for consensus up and down gene sets that were created for 30 mechanisms of actions of drugs profiled by the Lynx L1000 transcriptomics dataset. And then finally, the Aging Atlas has regulatory subnetworks inferred by ChEA-KG for up and down gene sets created from the GTEx data for 24 tissues. And these are just the QR codes pointing to each of these pages, but they're all available from the main ChEA-KG site. So now I'll demonstrate how we actually use ChEA-KG for a specific cancer-focused discovery project, specifically looking at subtypes of lung cancer. So for this use case, we're characterizing subtypes of the lung squamous cell carcinoma cohort from CPTAC 3, which has 108 tumors. So first, we identify clusters from the transcriptomics profiling of the tumors using multi-omics to targets. And then for each subtype cluster, we submit the upregulated genes to Enricher to identify enriched pathways. At the same time, we submit those upregulated genes also to ChEA-KG to identify the enriched transcription factor subnetworks. And then to characterize the transcription factors in each subnetwork, we submit each of those transcription factors to a new tool that was developed by our lab called GSFM. So GSFM, which was published last week in the Cell Journal Patterns, is a foundation model that's trained using over 1 million gene sets, and it can be used to reliably predict gene functions. So here we used GSFM to characterize the collective functions of each subnetwork for each cluster of patients. And then we merged these with the results from Enricher in order to characterize each subtype. So on the left, we have the enriched regulatory subnetworks for each cluster. And then on the right, we have the Enricher results as well as the predicted phenotypes for cluster 0. So based on the enrichment analysis results and the regulatory subnetwork, we can predict that cluster 0 is likely a hot cluster enriched for inflammation-related genes. And then we repeated this process for cluster 1 and found that several terms relating to cell cycle as a subtype is highly proliferative. So the subtype could be considered highly malignant and cold. And then for cluster 2, we identified several immune-related functions and phenotypes, suggesting this cluster is also an immune hot subtype. And then for cluster 3, we identified several terms relating to DNA replication, suggesting the subtype can be characterized as cold and highly proliferative. And then for cluster 4, we have several metabolic pathways enriched for the upregulated genes, suggesting that the subtype has significant metabolic reprogramming, which could potentially be targeted. And then finally, for cluster 5, there are several pathways relating to cilia structure and movement, suggesting higher potential for invasion and resistance. So, although the ChEA-KG subnetworks look like a depiction of a dynamic system, these are inferred for a specific molecular state, or in other words, a single time point. So, we realized that by applying ChEA-KG to time series data, this could be interesting because we can potentially trace the transitions of transcription factor activity over time. So, we developed an accompanying workflow for ChEA-KG called ChEA-KG time series, or ChEA-KG-TS for short. So, time series RNA-seq experiments characterize transient cellular responses, but they don't explain how those transitions occur. So, differential expression analysis from these data sets produces time-ordered up and down gene sets. And then these gene sets can be produced by comparing adjacent time points. You can look at time points versus the initial time point, or time equals 0, or compare perturbation samples to control samples at each of the time points. But in any case, the gene sets can then be used as input for transcription factor enrichment analysis tools. So ChEA-KG-TS is a workflow to investigate time series data sets. So we start with differentially expressed genes at each time point, and then submit those to ChEA-3 to identify the transcription factors that are controlling the transcriptional response at each time point. And then we can use the edges in the ChEA-KG background gene regulatory network to draw edges between the enriched transcription factors at each time point, potentially capturing the dynamics of the gene regulatory network over time. So here is a more thorough description of that workflow. So we start with a time series experiment, and then automatically compute up and down regulated genes at each time point, or you can provide pre-computed ones if you have that. And then these gene sets are submitted to ChEA-3 to produce the enriched transcription factors at each time point. These transcription factors are then fed to ChEA-KG to produce time series subnetworks. So the final output is a report that contains the enriched transcription factors visualized as a UMAP projection with the time point enriched transcription factors highlighted against all of the transcription factors in the background gene regulatory network. And then we also have these layered subnetworks of the enriched transcription factors and their connectivity across time points. And then finally, the enrichment analysis results for each of those transcription factors. So we applied ChEA-KG-TS to explore the response of triple-negative breast cancer to treatment with the cytokine trail. So this is using data from this study by Kundu et al., which aimed to unravel the effects of trail on the triple-negative breast cancer cell line, MDA-MB231. This is a figure from their paper kind of showing the schematic of the experiment. But trail is a possible therapeutic which has shown limited efficacy in clinical trials. But this study was actually able to show that trail induces NF-kappa B2 dependent cytokine production in this triple-negative breast cancer cell line. And they did this by performing time series RNA-seq transcriptomics after treatment with trail, measuring expression levels at 0, 1, 3, 6, 12, and 24 hours. So since this data set is publicly available in ChEA, we were able to get the RNA-seq counts and then analyze it further with ChEA-KG-TS. So here are the results from the analysis. So on the left, we have the time series regulatory subnetworks, where each row has the enriched transcription factors for each time point comparison. And so on the left side, we have blue nodes. These are the transcription factors that are enriched for the upregulated genes. And then on the right side, the red nodes are enriched for the downregulated genes. And then for the edges, the red edges represent inhibitory interactions, and then the green arrows represent activation interactions. So the transcription factors in hour 1 versus hour 0 and hour 3 versus hour 0 are enriched in well-known immediate early genes such as FOS and Jun. And then at later points, we confirm the involvement of NF-kappa B2 and REL-B. And so there is an interesting observation here. So NF-kB2 and REL-B are both inhibiting the same targets, Hive P1 and NFAT5, which are both implicated in NF-kappa B2 signaling. And there's actually a third transcription factor that's also inhibiting both of these targets, this ZNF267. So this suggests that ZNF267 is a novel member of the NF-kB2 NF-kappa B2 signaling pathway. And this is actually further supported by the UMAPs, which is showing ZNF267 clustering right near NF-kappa B2 and REL-B, suggesting a similar function. So ultimately, ChEA-KG is a new transcription factor enrichment analysis tool, which can be used to identify regulatory subnetworks given sets of differentially expressed genes. We demonstrated how ChEA-KG can be used as part of a workflow to characterize CPTAC cohort subtypes, specifically the lung squamous cell carcinoma cohort. And then we also showed ChEA-KG-TS, which is a workflow that extends ChEA-KG for time series analysis, and applied ChEA-KG-TS to uncover ZNF267 as a possible novel member of the NF-kappa B2 signaling pathway, which is activated in a triple-negative breast cancer cell line in response to the therapeutic cytokine trail. So ultimately, further experiments should focus on validating these experiments in vitro and also in vivo. But in the end, ChEA-KG and ChEA-KG-TS are ready to be applied across all CPTAC cohort datasets, and ChEA-KG-TS can also be applied to pseudotime single-cell datasets. So with that, I would like to thank everyone in the Myon Lab for their support and contributions. Especially thanks to Andrew Van Dusen, who is a summer student from MIT that joined our lab this last summer for a 10-week summer program and got the ChEA-KG-TS project started. And then thank you to Mehdi and the steering committee for the opportunity to present. And thank you also to Pei Wang and her lab, who is our PGDAC partner and the contact PI of our award. And then finally, here are the QR codes for both of the tools that I just mentioned, as well as our recent publication in nucleic acids research. And I will take any questions now.